Alrighty guys, my challenge is to keep you interested for the next 10 minutes. Let's see how we go. So, the paper I covered is Comparative Phosphoproteomic Analysis of Checkpoint Recovery Identifies New Regulators of the DNA Damage Response. It was accepted on the 13th of March 2013, was published in Science Signaling, has averaged about 51 issues a year, and an impact factor of about 6.3. The authors were So let's start with an outline. At first, I'll do an introduction, method, results, and then a conclusion to wrap it all up in the end. I'll start by putting this paper into context. So, what is DNA damage? DNA damage is an alteration in the chemical structure of DNA. The cell cycle or cell division cycle is a series of events that take place in a cell leading to division or duplication that promotes two daughter cells. Cell checkpoints are control mechanisms which ensure proper division of cells. Each of these serve a different purpose. Today we will focus on the G2 phase, which is a regulatory phase, checking for errors within the cell division. Cell arrest is the suspension of further growth in response to defects, i.e. DNA damage. P53 is a protein in the cell which suppresses most tumors and prevents mutation. It regulates DNA damage, and its inactivation or inhibition can cause serious harm. MDM2 is an oncoprotein which binds to P53 and blocks its function, promoting its degradation. Additionally, MDM2 and P53 have a negative feedback loop relationship. And as we all know, phosphorylation is the removal of a phosphate group from a protein. Small interfering RNAs are RNAs that interfere with the expression of specific genes with complementary nucleotide sequences. These cause mRNA to be broken down after transcription, resulting in no translation. Astrin is a mitotic spindle-associated protein expressed in all human cell lines and tissues. Kinases are a type of enzyme that catalyzes the transfer of phosphate groups from high-energy, phosphate-donating molecules to substrates. Now, I will outline how the experiment was set out. They started off with explaining how cells recover from DNA damage, when a G2 cell encounters DNA damage, what happened once the DNA damage was repaired, and how they performed large-scale quantitative phosphoproteomic analysis and identified changes in phosphorylation. So now I'll outline the method. Cell culture, transfection, and drugs is when they used Astrin-inducible cell lines treated with caffeine to enter mitosis phase. Plasmids and oligonucleotides of human astrin with complementary DNA was purchased from Origene and converted into an nRNA-resistant mutant SO41A by site-directed mutagenesis. They brought antibodies against P53, MDM2, histone H3, clasp1, and astrin. For the phosphopeptide enrichment and mass spec analysis, 35 fractions were collected, and an offline chromatography was used to enrich further selected fractions. They used a nano LC LTQ quadritrap. For the peptide identification and quantitative analysis, they used a Mascot V2.2 search engine against a SwissProt 5.6.2 version, with an FDR determined as less than 1%. They created an siRNA library composed of 4 on-target duplexes per gene, with siRNAs targeting P53, SCAP, GAPDH, and mRNAs were on target for smart pools of damomoxicon. Here we can see an outline of how they cultured their two different cell lines, with ones with induced DNA damage and others without any DNA damage. The use of caffeine was heavily used as a treatment. Here we can see a rough outline of how they extracted their two different cell lines and then uploaded them into an Orbitrap for mass spec. So now, let's move into the results. This will be broken up into groups, starting with comparative phosphoproteomic analysis of checkpoint recovery, Astrin and G2 checkpoint recovery, the role of Astrin in the proper activation of p53, So, they found 715 differently phosphorylated proteins, with a large proportion having kinase and phosphate activity. 84 from the 715 were identified in previous proteomic screens for ATM and ATR. This indicates that proteins modified by DNA damage, responsive kinases, are modified once the kinases are inactivated. Additionally, they found that commercial antibodies confirmed decreases in phosphorylated clasp 1 at VH656, and this helped to confirm their results further. They found 28 proteins differently phosphorylated across all time points, and within 2/3 of their experiments they validated 10 proteins as DNA damage regulators. Thus, their proteomics-directed sRNA library screening was a success with a high hit rate. In this diagram, you can see the number of single siRNAs, which had in the top 10 on the left, with the asterisks above each. These had a high hit rate of just below roughly 80%. They also found 11 potential Astrin interactors in the cultures treated with deoxyribosine. This included SCAP, a known partner of Astrin, and verified phosphorylation of Astrin at 041. This was then reduced with PLK1 inhibition, thus PLK1 phosphorylates Astrin on 041 in vivo. They then found that Astrin itself may be a target of p53, as their data suggested that the deletion of Astrin leads to transcriptional repression by 53 and equals a reduced ability to recover from G2 arrest. They suggest that the regulation of p53 by Astrin is at the post-translational level. They also suggest that MDM2 is a dependent phosphate degradation of p53 and is impaired in all cells that lack Astrin. Also, they conclude that Astrin itself may suppress p53 by promoting MDM2 depletion and degradation, as it is known that MDM2 and p53 work in a negative feedback loop manner. So, in conclusion, they performed phosphoproteomic screens and found 700 different phosphorylated proteins. 154 of these were reproducible, and therefore large changes do occur as cells silence DNA damage checkpoints to re-enter the cell cycle. Also, they found that PLK1 controls recovery through the reactivation of cyclin and CDK. This result suggests that the inactivation of proteins is not just regulated through dephosphorylation, but might also involve phosphorylation-dependent inactivation or degradation. Therefore, PLK1 has a role in silencing DNA damage checkpoints and the activation of cell cycle recovery and re-entry machinery, and that both of these aspects of recovery are strongly connected. They also proved that their high hit rate was a combination of the shotgun proteomics plus the siRNA-based screens they performed, and that this gave an excellent method to recover new regulators of given cellular processes. They conclude that aspirin is required for mitotic re-entry after DNA damage is induced and due to arrest, and its phosphorylation by BLK1 at SURO4 is important to its function. Also, aspirin-deficient cells have reduced MDM2 activity, which therefore affects p53 as they are in a negative feedback loop. Further studies are needed to elucidate how aspirin regulates the abundance of MDM2. Therefore, it can be concluded that the dataset that they've provided can be used for future reference, and that their strategy developed can easily be applied to the analysis of other signaling networks in different cellular processes. Finally, I will finish with a review of this paper. The paper was great. The points were clear and set out. It was only roughly about 8 pages long. It's been cited 5 times, and it deserved to be published because it has brought forward new insight into how the cell cycle regulates and works, and new potential methods and analyses techniques for future work. Thank you for listening. For further information, please refer to my references.